Okay, I'd like to welcome everybody to the April 21st meeting of the working group. I'm very pleased that we have two excellent speakers who've agreed to talk to us today. As always, I need to remind you that the meeting is live streamed and recorded with slight editing for later viewing. So please do be aware of that. Both of our speakers have agreed to stay if there's interesting discussion after the main talks. So we'll have two 25-minute talks followed by a half an hour or so of open discussion. As always, Reinhard and myself are available for comments and questions. Reinhard is traveling today. He very rarely misses these meetings. He'll watch them on recording afterwards. He's going to Puerto Rico. Jim Sluka and Lorenzo are also always here helping us. Jim is also out today. He hopes to be back next week. Please feel free to contact us with any ideas, suggestions, comments. Please help us publicize the working group meetings. Again, we have our great set of YouTube archive talks. We have a Twitter feed. You could put MSM Viral in your feeds. Remember to tag things. Slack channel. Please do send us your papers and results that are coming out of things related to the working group. We have a pretty impressive list of papers related to working group activities that we put up on the IMAG MSM wiki page. And we definitely would appreciate your helping us keep up to date on that. If you have things like faculty positions, grant applications, postdoctoral openings, graduate student openings that you would like to have advertised to the community, Lorenzo could certainly help us distribute those. So please take advantage of this community. It's not just a seminar series. Again, we have the Twitter feed, MSM Viral. We're trying to build that up a little bit to build our audience and to make sure that we have an identity. Are there any announcements before we get started with the main meeting today?
This is Reed. I can just make a quick comment that there's a NIAID workshop coming up in May. It's about systems biology with a focus on vector-borne diseases, and I'll put information in the chat.
Great, that would be great. Thank you, Reed. We always appreciate hearing about conferences and grant opportunities from NIH. It's very kind of you to be such a supporter.
Sure,
we will have our digital twins subgroup meeting tomorrow morning at 11 a.m. Reinhard has promised us that he'll be available to chair that. As you know, that's an exciting development coming out of the working group. There are going to be two conferences coming up, one in the fall and then one next summer, led by Reinhard and then by Reinhard and Anna, that are going to be quite impressive. We're currently trying to write a proposal for a third working group, a digital twins meeting to be held in Leiden. And a lot of you have been helping with that. So if you want to be part of the Digital Twins work subgroup for tomorrow, and you need a link for that, let Bruce know, and he'll be happy to send you the link. And we definitely appreciate your participation on that. It's not replacing the main working group, but it's certainly very complementary to that, and the expertise is very comparable. Any other announcements before we move on to our talks? Okay. For upcoming meetings, next week we have two talks coming up. May 12th, Joy has agreed to talk. May 19th, we have Chang Zhang. We don't have anybody yet for May 5th, although we have a lead for a speaker. So please help us keep the schedule full. There's no problem having a single speaker in a week. But please suggest yourselves, your friends, the papers that you've read recently that you like. Feel free to come back and talk to us another time. Remember that we have a pretty wide remit. We are happy to have talks on modeling, on the immune system, on viruses, on epidemiology, on experimental techniques, on computational techniques, on clinical applications, on industry. We've had people from all sorts of fields, so please don't be shy about suggesting things, even if you think they might not be a perfect fit. And do feel free to put your graduate students and postdocs forward here. We have a lot of very distinguished senior speakers, but we've also had some superb talks from junior scientists, and this is a good opportunity for them. So please do help us keep the agenda full. Lorenzo does an incredible job of reaching out and securing speakers. So please help us out. So without more ado, I will move on to our first talk today. We have Daniel Reeves from Fred Hutchinson, who's going to be talking to us about SARS-CoV-2 evolutionary dynamics. And I will turn it over to you.
Okay, great. Well, thank you very much for the invite and for Lorenzo for coordinating. I got the sort of story that we're looking for like a 15-20 minute talk, high level, looking for collaboration. So that's sort of what I've done today, and just happy to dive into details later if anyone wants to do that or stop me and so forth. So the talk title I gave was about a paper that we just published about multi-scale modeling and super spreading events for variants of SARS-CoV-2. Just a note, I mean, I'm an associate in vid, which is sort of like a junior faculty terminal postdoc purgatory type position. And I'm at the Fred Hutch in Seattle, and my background, I did a PhD in physics, but I now do infectious disease modeling. So just to start this, the things I really like and the things I think about all the time is viral dynamics usually. And I think recently I've been thinking a lot about how to fit models well and how to choose the best model. So if anyone's interested in talking about that, I'd be happy to. I've been using nonlinear mixed effects modeling with this free software Monolix that I really recommend. It works amazingly well. And we've published on, at least in COVID-19, epidemiological models, and I think particularly interested in what types of vaccines can do different things to the population. So we're really interested in whether vaccines completely prevent infection versus blocking symptoms and so forth. What does that do to the epidemiological dynamics? I have a sort of personal interest in how to analyze big complex models at this point, and how do you learn the most from them, given that intuition kind of fades for me sometimes. And particularly thinking about evolutionary dynamics within hosts is the sort of main thrust of my program. One thing I present today a bunch of computational and sort of simulation results, but I am really interested in if any of you have thoughts on what kind of analytical stuff could be added to this story as far as super spreading in over-dispersed networks. Okay, I'll begin with acknowledgements. A lot of this work was done by Ashish Goyal, who's just an amazing scientist who did about 3 PhD theses worth of work in the last couple of years, I think, and now is at Pfizer. And Josh has been a great mentor to me for years. Other people that helped with this project, Fabian, Brian, and Liz, all at the Hutch, and other people here, and funding from the NIH and the Council of State and Territorial Epidemiologists was helpful for this. Okay, so the story here starts, I guess, with the beginning of finding viral loads in people that had SARS-CoV-2 and building a within-host model for this. So this is from Ashish and Fabian's paper in 2020. They have this viral dynamics model, and just to point out, it fits really well, but there are other within-host models from a bunch of different groups that also fit similar data well. And I think a trend through all of this is trying to decide how complex of a model do you really need or want. And in this kind of data, how do you deal with this fact that a lot of the time the peak viral load was missed? There's some people where you see this sort of classic trajectory of up, down, flat, but others, many peaks missed. And I think this is particularly where that population nonlinear mixed effects modeling helps you borrow strength from some of these great data sets and project onto the less sampled ones. So just a quick aside on a modeling term that's in this and also in other viral dynamics models is a sort of power law term. So I'm just showing you a couple panels of nice fits to good HIV viral load data. And this model is three equations, three differential equations, but it has this term where the number of infected cells is to a power. And just to highlight, this is kind of a tricky term. It really makes fitting work. It's really flexible and you get really heterogeneous trajectories. But I've been curious what this really means in terms of thinking about model selection, because you've really only added one parameter, but it's in a different way than all these other parameters. The fact that it's not a nonlinear parameter, or sorry, it's not like a multiplicative parameter. It does something interesting. So if anyone has thoughts on that, I'd love to talk about that. Okay, so back to the results here of just SARS-CoV-2 within hosts. I think the first thing that that was shown here is that given the viral loads, how they look, it's really hard to treat somebody post infection. So this is just a viral load trajectory of what happens if you give really good therapy, like the green one, but at day 5, you still have a ton of area under the curve here, and most of the transmission potential has already happened. If you give it, like on the first day or something, you can minimize the infection, but this is a very hard challenge given how many SARS-CoV-2 infections are asymptomatic. So it's post-exposure prophylaxis still seems worth it to try, but this was just a kind of unfortunate wake-up call at the beginning of how hard this was going to be. And then from there, we moved on to epidemiology. And so it's known for a long time that SARS itself had a very high super spreading potential. So this is a graph from a famous paper from Lloyd Smith et al. But in terms of the most infectious cases, how much transmission happens, it's sort of the Pareto 80-20 principle. It's already really happening for SARS-1. And so Ashish, again, in another paper with us, has shown that if you compare flu and SARS-CoV-2 viral kinetics, they don't look that different, but there's a lot more super spreading for SARS-CoV-2. So this kind of suggests that there's different transmission modality, and perhaps I think this was an early note that there's more aerosolization or something, or the super spreading was just going to happen more commonly. So we now have this multi-scale model that captures a bunch of these things at once. So it has sort of viral loads built into it. There's individuals that have different viral load trajectories. There's a dose response transmission probability. So you sort of draw a viral load and then say, okay, if you saw somebody on that day when you had peak viral load, how likely would it be to actually transmit to them? And then you draw a distribution of contacts. So there's like, a person sees one person on one day and then maybe sees 8 people on another day. And this can be a very over-dispersed network. But it is not completely sort of a network framework in the sense that the same person doesn't have a super spreading potential, so to speak. All people are drawn from the same kind of network. And then the new thing we added for this paper as well is just that every once in a while, there's a new variant created. So after an infection, there's some probability that you get a new variant. So you put this into a sort of epidemiological model where there's a bunch of susceptible people. You introduce a single person with a variant, and then the model output looks like this, where you have variant incidence and then time after the initiation of the epidemic. And in particular, we were really interested in tracking super spreading events, which we define as more than 5 secondary infections. So just like a first result is that, like a sort of classic model with over-dispersion, extinction is more likely, and you can still have extinction of situations where the variant reproductive number is high. And in particular, if there's only one individual, this color bar tells you the percent of these simulations that were going extinct. So with low reproductive number, yellow, very high probability of extinction. But even with higher reproductive numbers, still decently high chance of extinction, and particularly as you make a model with more and more over-dispersion, this remains true. So this gets stamped out a little bit if you get to more initial cases. So the initial conditions definitely matter as well. And the conclusion we took from this, again, I guess for the sake of this talk, for discussion fun, I'll be a little provocative, but I think one of the things this suggests is that dangerous variants of SARS-CoV-2 or other coronaviruses with pandemic potential could be getting introduced, but going extinct a lot more than we think in the population. And it takes some minimal threshold of 10 or 100 or something to start making it more likely that this extinction will not happen. So I think this really justifies preemptive planning. Just a note on that is that the reproductive number is not like independently changed in terms of the dispersion parameter, so that you really can see that the number of contacts matters for the reproductive number, but the dispersion parameter doesn't necessarily change these colors. The next result really is that the viral load also matters. And so the plots here again is like starting with 1 initial case, 10 or 100 initial cases. The x-axis is now the percent of simulations resulting in extinction. So, and then the y-axis is the peak viral load. So sort of averaged over everybody in the simulation. And what you can see here is that for a single initial case, there's still a lot of simulations that result in extinction, but those all happen with low average viral loads. So sort of if variants were not as good, they didn't have as high viral loads, they're still much more likely to go extinct in a low initial case situation. And so I think that's actually rationale going back to trying post-exposure prophylaxis, because if you can even lower the peak viral load a little bit, you can really break down these chains of an infection from a decent growing variant. Okay, the next story is that if you put this all together in time, the timing of the variants, the stochastic process also really matters for what ends up winning. So these simulations are starting with a variant that's at a baseline and has a effective reproductive number of 1, so it's staying flat in the population, and then introducing new variants randomly throughout this that have different reproductive numbers by the color. And so this is 9 simulations. There's like thousands of these, so sort of summarized in the paper. But just looking through these, I think the key is that the invading variant isn't always necessarily the one with the highest reproductive number. Sometimes it is, like this pink one is winning in some of these simulations, but there is other situations where a variant that's not even that much better than the one that was around wins because it was there first, and then it becomes really, really hard for yet another variant to come and take over again. So there's sort of this by chanceness of which variant ends up winning is, I think, also important for thinking about epidemiological dynamics and thinking about whether or not you're out of the woods, so to speak, if you get a variant by chance that happens to be a lower reproductive number. A lot of this makes me think too, in terms of thinking about viral load, about rapid testing. And so I think by now, I actually got to go to a conference for the first time a few weeks ago. And so everyone was taking these rapid tests the whole time. And I've seen some pictures now of rapid tests that have sort of partial color. And I think it's really important that we start thinking about whether the high viral load as detected by PCR versus rapid test is relevant. So I just put a couple things on here. The first is some work we did for HIV, where there was this study done where there was a sort of rapid test like thing being used, but it turned out its value was actually correlating really well to HIV viral load, suggesting that test was actually a decent measurement of sort of quantitative measure as opposed to just qualitative. And now for SARS-CoV-2, I just found this medRxiv paper, but they're showing that depending on viral load, it's across different tests. Lumira, BD is some other brand. So a bunch of different brands. You get positive tests in a distribution and negative tests in a distribution. It doesn't mean some of these people didn't have any viral load, but just that it was lower. So I think in terms of thinking about this link between transmissibility at high viral loads and this pragmatic balance of, do you want to keep people quarantined even when they have low sort of simmering viral loads is another sort of provocative question worth discussing. Jumping back into this last sort of slide of data from the paper, the early super spreader event happens can really predict invasion. So kind of the same story here as that invasion slide, but quantified a little bit, which shows you that the day of the first super spreading event for a variant is strongly predictive of how many days it takes for the variant to ultimately invade. So this is less true for weaker variants with reproductive numbers around, closer to 1. But there's a strong association with an early super spreading event, meaning more than 5 secondary cases, and then a variant eventually coming to take hold of the population. That isn't true in terms of the number of super spreading events. So that is a super spreading event later in the chain of infection is much less important than the early one in terms of the variant ultimately coming to dominance. So the invading variant reproductive number does matter. Of course, you see that here, but this relationship holds through a lot of the spectrum. Okay, the summary here, I'll check what time it is. Is that the SARS-CoV-2 kinetics are really rapid, and the frequent asymptomatic infection poses big challenges for antiviral therapies after infection. However, reducing viral load even slightly on average could be important for breaking chain of infections of new variants as they come to rise. The comparison of viral dynamics, I think, within hosts can actually help inform epidemiological processes. That was a cool thing we found here, which is that if the viral loads look the same, but the spreading dynamics are really different, that probably says something about some functional form of contact patterns. And the third point is that the dangerous variants are probably frequently created, but they just go extinct because of this huge over-dispersion parameter and reproductive numbers that are near one, can be above one, but still going extinct. And finally, the super spreading events, particularly early after a variant is created, is a likely determinant of whether that variant eventually emerges. Okay, so the next chapter here is how is sort of where I'm going with this next. And so if the super spreading event is really crucial for the variant emergence, I think it's still really important and exciting to think about what the biological basis of these new variants are. So 3 hypotheses on here. Definitely let me know if you think of more of these or you have a list yourself. But so there could be zoonotic reservoirs. This has been now documented in white-tailed deer transmission in a couple different papers. So there could be circulation among deer, and that could be creating new variants and back to humans, sort of as in influenza or something. There could be human epidemiological reservoirs. This is harder to reconcile, but there could be places where people are infecting one another and evolution is happening, but this is not being detected on the global scale. And I think probably the favorite hypothesis at this point is that, unfortunately, some of this evolution is happening in people who are immune compromised and have very long viral replication shedding patterns, and sometimes I think failed treatment. So I say, is this really worth modeling? And I think so. So just to show you some data about this, because I think it's really fascinating. There's 2 different studies of this. This one's in New England Journal, this one's in Nature, but this is a graph of a bunch of different global SARS-CoV-2 patterns. And this red part of the tree is from a single individual. So you can see that this person who had prolonged viral shedding, this is their sort of clinical trajectory over hundreds of days, and they had some treatment at one point, so their viral load went down, but then it came back, and then another treatment. So it's a complicated course of infection, but they had more evolution in that 100 days in terms of number of base pairs different than most of the other evolution across the entire tree of people. This is within a single person getting a lot of base pair differences. And you see that same story in this paper too, where there's this person that had a very, very extensive amount of diversification and divergence of their SARS-CoV-2 virus compared to a bunch of other people, and even when compared to people who had prolonged shedding patterns. This is like this blue one, this pink one, and this red one. Okay, so this brings me to some recent work I did on HIV that I'd really like to now integrate into thinking about SARS-CoV-2. But this is a model we just published on within host phylogenetic. So I call this the Wi-Fi model, but this is a viral load where you track the viral load total over time, but broken up into a big multi-strain model. And what you can do from there is you can compute the genealogy, or sort of like the true tree. You have access to which strain gave birth to which other strain. It's basically an agent-based model in this sense. So you know who the parents are the whole time, and you can compute the whole genealogy of the whole infection. And then furthermore, you can kind of downsample this to look like a realistic sampling trajectory, and then build a phylogenetic tree using a typical Bayesian analysis and compare this to experimental data. So this is the sort of direction I'm going next, and I'm excited to maybe talk more about that too. So I'll stop there and thank you all for your time and any questions at all.
Okay, thank you for covering an amazing amount of material in a short amount of time. We'll go on to our second talk and then come back. So I hope people have written down questions. I've certainly got quite a few, and appreciate very much the presentation. So we'll move on to our second talk for today, which will be on quantitative prediction of conditional vulnerabilities in regulatory and metabolic networks. And we're moving from viral infections to bacterial infections. So it'll be a nice change of pace. Please take it away.
Thank you. All right, well, thanks for the invitation. So yeah, I'll speak about strategies that are needed to address the rise in antimicrobial resistance, and thank some people first. I'll acknowledge them as I go, but just wanted to quickly put their faces and the lab names here. Also funding from NIH, NIAID in particular, NSF and the Gates Foundation. So very quickly to put things into perspective, we know that antimicrobial resistance is on the rise and is projected to cause up to 10 million fatalities by 2050, and a quarter of those are believed to be because of tuberculosis. And to put TB into perspective, infection by a sensitive strain of TB takes just about 4 drugs over 6 months to cure about 95% of the patients. But if the infection is from a resistant strain, then you need about a dozen pills administered over 30 months or so. And that's about 15,000 pills, and still only 56% are cured. So the reason why TB is so successful in evading antimicrobial therapy is because of the heterogeneity that it can manifest in its physiological states depending on the microenvironments. And this is just a snapshot of a granuloma that shows that the bacillus shown in red here can live in a variety of different host environments within host cells of different lineages, and in later stages of the disease also extracellularly in the caseum. And across all of these microenvironments, it experiences very different conditions, and it has evolved the capability to adopt heterogeneous states that have very different drug susceptibility profiles. And we don't really understand a lot about these heterogeneous states, and we would need to understand it at a causal and mechanistic level so we can then begin to predict what are the vulnerabilities in those states and how we can target them rationally with drug combinations. And what we need are combinations of drugs that can potently kill the heterogeneous subpopulations, and also these drugs need to be synergistic with each other, and they should not cancel each other's action. This is even more important in cases of co-infection with HIV, because drugs used to treat TB can interact with HIV drugs and negate the effects or increase toxicity. So how does one go about doing this? And we've developed a framework of tools, and I'll just show you 2 today. And the first one is called EGRIN. It's a model for gene regulatory networks, and I'll show you how we build the models. But in the case of TB, we need to generate multi-omics data sets for the heterogeneous states of TB. So we built a series of technologies, and I'll list a few. This is a technology to isolate dormant non-replicating cells called persisters that can be generated stochastically in actively growing cultures. And we can then profile these subpopulations with technologies like RNA-seq. We've also built reactors that can simulate host-relevant microenvironment changes, such as in oxygen tension, and slow down the transition so we can capture the early, mid, and late phases of the response, and the transient phases especially, to get at some of the mechanistic drivers of the state transitions. And we've also developed technologies to enrich pathogen RNA from infected tissues, so we can profile them in quantitative ways, so we can then begin to dissect the networks that are responsible for the states that they adopt inside cells. Now, we are not the only ones who are generating these data sets. There's a lot of work that's ongoing. And we've compiled data from a large number of laboratories, 30 laboratories in particular, roughly 53 studies. And all of these studies, essentially, including ours, are probing the response of the pathogen to a range of different variables that are relevant to the infection cycle, be it from the host or from treatment, and also in the context of different phenotypes that they elicit. And these compendia of transcriptome changes are then analyzed through a inference procedure that we developed. It's done in 2 steps. So first we do a biclustering, and it's actually multi-clustering, where we use an algorithm called CMonkey to identify which genes are co-regulated in what context. And we guide that discovery process with networks of prior information, such as gene association through evolution, or in metabolism, shared cis-regulatory elements and promoter elements, and so on. And we identify modules of genes that have similar expression patterns that are supported by shared regulatory mechanisms. So they're co-regulated and not just co-expressed. And this is just one module that we've discovered here in this example. And we can then take this individual module and apply generalized linear regression to identify what regulators are responsible for driving the observed expression changes. And this is a predictive model that can be used to then predict what would be the response of these genes in these modules across the experiments that were used to generate the model, and even in new experiments. And I'm showing you measured versus predicted in red and blue respectively. And the boxed region is a set of experiments that were from a test set. So there were new experiments done after the model was constructed. And we've shown that this model is quite accurate in predicting new responses. And as I said, this is for one module. We can uncover the entire regulatory network for all genes in the cell. And there's a lot of papers I've listed here above that report various aspects of the model inference. Then the model, it can be used to do 2 things. It can be used to explain all the behaviors that were observed in the experiments that were conducted, and also predict responses to new environments and new genetic perturbations. Now, this is a model that is based on a biclustering step. So that's an NP-complete problem, meaning we can't really get a deterministic solution. The heuristic solution approximates what the exact composition of each module would be. So if you run this algorithm multiple times, you'll get approximations of the solution, but not always the exact solution. So we've built an ensemble-based approach to address that issue. And essentially, we run the whole procedure multiple times, hundreds to thousands of times. And that generates a space of associations amongst genes and the context in which they're co-regulated. And in this example here, I'm showing you a set of genes. In the center here are co-regulated with genes in the yellow Venn diagram in conditions shown here to the left. And those genes break away and become co-regulated with a different set of genes in a different set of contexts. And we can even predict what are the mechanisms responsible for dynamically breaking these clusters and bringing them together with the other clusters. And here's a view of what that looks like. So this was done with a different organism some time ago. And we're looking at how the genes get co-regulated, and co-regulation is shown in colored edges. And genes that are up are in yellow or in blue and down-regulated. And this is in response to gamma radiation. When we shift to a new condition, we find that completely new regulatory associations emerge and change the whole modular architecture of the network. And in comparing the 2 states, we can then identify what are the regulatory states that are associated with adaptation to each of these 2 conditions. And in particular, what are the regulatory mechanisms that are unique or shared across the 2 conditions. And so in the context of TB, we've built this model, which we call EGRIN 2. And as you can see, it covers a large number of modules because of the ensemble runs that can then collapse into what we call core-REMs, or co-regulated modules. And they're associated with these gene regulatory elements. These are cis-acting promoter elements that we can locate across the genome, even in non-coding regions. So we can find some novel regulatory mechanisms. And importantly, what we can do is, because of this post-processing, we can now identify context-specific regulation. And I'm just showing you examples of some conditions such as osmotic stress or low ion or acidic pH, which are present in small numbers in the overall transcriptome compendium. And by definition, in one single run, we would not have the statistical power to find regulatory mechanisms in those contexts. But when we run this ensemble-based approach, we begin to identify modules that are conditionally active in those contexts. And these contexts turn out to be really important and typically conditions that are difficult to interrogate because of the nature of the slow growth of Mycobacterium tuberculosis, having to grow it in the BSL-3 conditions and profiling in host intracellular context. So this approach really allows us to then increase the statistical power and boost our signal of mechanisms that are operating in rare contexts. In particular, I'll show you an example of a mechanism that is operating in low pH conditions, which is a condition that is really important in the infection cycle of TB when it goes inside a phagosome that then becomes acidic, so the lysosome can merge and kill the cells. But MTB has adapted to this shift in pH, and we've discovered a key network that is responsible for this adaptation. And in particular, what we found was there's a particular corem 71 that is conditionally active in acidic pH. So you can see association with pH here, and that is regulated by this gene here, which is MTRA. And we also implicated the mechanism by which MTRA regulates genes within this corem. And the genes within the corem are a collection of amidases and hydrolases that are associated with cell division, control of cell division. And the inference procedure allowed us to get down to nucleotide level resolution, and we were able to recapitulate the binding site of the transcription factor, and that correlated beautifully. In fact, had better signal than the motif that was discovered through ChIP-seq or more targeted approaches that were previously reported. And what this model predicted was that in low pH context, you would have higher fitness consequences of perturbing the network. And I won't show you the data, but we did T and seq analysis, and ELISA showed that this particular set of genes do become conditionally more important in low pH conditions. And in fact, because of the functional enrichment of cell division in this corem, we were then able to validate using some more detailed characterization that if you knock down the regulator, and we did this with an inducible system, CRISPR-I system, it inhibits cell division, and then the cell length increases significantly. In fact, cells don't divide. They get this really strange clubbing, like hockey stick-like appearance with many septae. And furthermore, we also went on to validate that when you knock down this regulator, the Mycobacterium tuberculosis cells become even more susceptible to isoniazid and rifampicin, and you get several logs killing compared to just in the absence of CRISPR-I induction. So this is an example of how we can use the ensemble modeling at the regulatory network level to find context-specific vulnerabilities that can be targeted in a very rational way. Now, a lot of the transcriptional changes cannot be easily interpreted as to what consequences they might have. So we developed a second algorithm called PRIME. And this allows us to then take the weighted regulatory influences that are discovered using the EGRIN approach, and then model the consequences of those influences on metabolic genes that encode enzymes within metabolic networks. And in so doing, we can now predict growth phenotypes. And for example, here I'm showing you the predicted effects of drugs on the growth rate of MTB as compared to the measured growth rate. And you see it's almost a perfect correlation. And we even contextualized the model for intracellular context by taking transcriptome profiles of the pathogen from within macrophages of different lineages. And we essentially built on work done by Bernard Paulson's group over a decade ago, and setting interfacial constraints and then using expression profiles of the pathogen from different host lineages, we were then able to build alveolar macrophage specific models, monocyte-derived macrophage specific models, and bone marrow-derived macrophage models. And these models allowed us to then simulate and predict growth characteristics in each of those host cell lineages. And these matched up beautifully with experimentally measured growth phenotypes of the pathogen in each of these contexts. And essentially what I'm showing you is the range of growth characteristics, growth rates that are possible in each of the host cell lineages. And as it's well known, the monocyte-derived macrophage is repressive or restrictive for growth of the pathogen, whereas alveolar macrophages are more permissive. And in broad conditions, you get a much wider range of growth rates that are possible. It essentially tells you that it doesn't really simulate the host cell specific phenotypes and essentially gives you a range of growth phenotypes based on availability of oxygen and other resources and not necessarily the host adapted context. Now, because the model is causal and mechanistic, for the first time using this model, we can now predict the consequences of knocking out regulators on growth characteristics in a context-specific manner. And in the past, using metabolic models, you could just make predictions of metabolic genes. And people have made some attempts at predicting effects of transcription factor knockouts with marginal success, and that includes our prior work as well. But now our predictions are really accurate and quantitative. And here I'm giving you one example from a genome-wide fitness screen where we, using PRIME, predicted the consequences of knocking out transcription factors and rank ordered the fitness consequences based on each transcription factor and compared it to experimentally determined rank orders. And they're statistically very significant or very similar. Now, in comparison to prior efforts, as I mentioned, there were two in particular, and before that, there was one more called RFBA, which did not really use a regulatory network, just used expression data. But PROM was one of the first attempts by Nathan Price and his group to predict the effects of transcription factor knockouts. And here they used just a protein-DNA interaction network that was generated in one condition and expression data to predict the consequence of knocking out transcription factors. And this was a probabilistic approach. It would just tell you probabilistically whether knocking out a transcription factor would block flux through specific reactions. iDream was a advancement over that. Now, iDream took advantage of the EGRIN model, and we worked with Nathan to build this model. It was definitely an improvement, but here we did not really take advantage of the weighted regulatory influences. We just took the regulatory influences and converted that to false discovery rates, essentially to predict whether transcription factor would regulate a gene or not. So the dotted line shows these regression-based influences that were uncovered with EGRIN, and the solid lines are with ChIP-seq. So the network certainly improved, and you can see on the right in the bar graph that it improved the coverage of the model. Now with PRIME, we went even further and we did several advancements. The key ones being, we took advantage of the weighted regulatory influences. We also took advantage of the knowledge of combinatorial regulation and the level of individual genes, and then also the level of reactions. And the coverage looks similar to that of iDream, but the performance is way better, right? So in each of these approaches, you're in the end predicting how transcriptional changes manifest in changes in metabolic flux, and thereon using objective functions, you can predict different phenotype consequences. And you can see with the PRIME, you can predict quantitative flux changes, not just whether flux can happen or not through a given set of reactions. And here's a couple of validations we did. So this is the network of MTB that is essential for growth on glycerol, which is the carbon source on it, which grows prolifically. And I'm just showing you reactions that are essential, shown in black. Gray are reactions that are not essential, while growing in, or I should say, genes that encode enzymes in reactions that are essential. And on the very top are transcription factors that control those genes. And this is for glycerol, but when you shift to cholesterol, the network obviously changes, and we identify which regulators are now become more essential and what are the essential metabolic genes they control. And using TN-seq data for cholesterol and glycerol growth, we then did leave one out cross-validation analysis to calculate the accuracy of prime as compared to prom and iDream. And across both these contexts, prime outcompeted both of those algorithms. So in a sense, I think we have an algorithm that is significantly more accurate and quantitative in predicting phenotypes and consequences of targeting metabolic and regulatory genes in specific contexts. I'll give you one example, one highlight of how we can use this to characterize specific vulnerabilities. And using EGRIN, originally we identified a novel regulator called MADR for mycolic acid desaturase regulator, which controls two desaturases that are part of the FAS2 complex. And these are involved in desaturation of mycolates. And as you know, mycolic acids are this special waxy lipids that give a lot of protection to mycobacteria from toxins and drugs and so on. Now, when there's stressful conditions, this regulator, which can exist in two forms, a cofactor bound form or a free form, and depending on whether cofactor is bound or not, we had predicted that it would either be bound to DNA or not. And so when it infects the host cells or when it is going into a low oxygen condition, this regulator comes off. It's a repressor, and this allows the desaturases to be overexpressed, and that leads to remodeling of the cell wall, putatively allowing the pathogen to resist the intracellular challenges from the host. And then as it advances through the infection process or adapts to stress, it then shifts back to the DNA-bound form, which then represses the expression of desaturases. And this is when the pathogen has gone into a dormant, non-replicating state. So now here we had actually predicted that the cofactor bound, there's a cofactor that binds, we didn't know what it was, but the model suggested there's a cofactor, and we thought the cofactor bound form was the DNA bound state of MADR, but we just published a PNAS paper, and it's actually the opposite. Turns out MADR binds to saturated acyl-CoAs of length C16 or greater, and when it detects those, it comes off, so it can then express the desaturases that can go and then do the desaturation of those long-chain fatty acids. Now, if you knock down the regulator, you get several log killing of mycobacterium. And you can see here on the left is when you don't induce the CRISPR system, there's no effect. But when you induce it, you see several logs killing. And using prime, we then analyzed MADR in some more detail, and it suggested a more systems level effect that would manifest in susceptibility to multiple antibiotics and not others. And it predicted 5 antibiotics that would be potentiated by MADR knockdown. And we confirmed that with kill curves, and it predicted some antibiotics would have no effect, and we confirmed that as well. So this again shows how we can use prime to get at context-specific vulnerabilities, and also it's very specific to the context in which the vulnerability manifests. So this allows us to now think more broadly at a systems level about how we can use this multi-scale modeling to understand how the pathogen transitions from susceptible states to more tolerant, dormant, non-replicating type states like persister-like states. And we've started already identifying the networks that drive these transitions. And within those networks, we're identifying vulnerabilities that can be targeted with drugs that already exist or new drugs. And these could be drugs that target steady states or transition states. We're also looking at using this approach to understand how the pathogen gains resistance to the antibiotic, because eventually few cells might escape and gain resistance, but those are trajectories that could also be mapped, and we can identify vulnerabilities in those trajectories and block them. And we've also done work now to figure out novel states that are adopted by these bacteria when they gain resistance, so we can then find new vulnerabilities that can be targeted to reverse resistance. So there's a variety of different ways we can now think of using this approach to rationally formulate these treatment regimens. And if there's time, I can just very quickly give a example of how we've also applied this to C. difficile infections. Is that okay? Or this is one more slide? Yeah. So this is work we did with Lynn Bree, and also she's at Harvard, and we also collaborated with Bruno Dupuy in Pasteur Institute. And what Lynn had established was that when you co-colonize mice with commensals, they can protect or worsen the outcomes of C. difficile infections. So in particular, if you co-infected with Clostridioides sardiniensis, then you would get a potentiation of the infection, which is shown here in the red curve here. Whereas if you have co-colonization by Paraclostridioides by fermentans or PBI, then you would have protection. And the question was why? So we built a prime model, EGRIN and prime models for C. diff in various contexts. And these are published in 2 cell host and microbe papers that were published back-to-back last year. And using the model, we essentially uncovered the networks that were active in monocolonized or co-colonized contexts. It's very, very busy here, but just to tell you the systems level perspective we can get, even for an intracellular context from mouse gut. And we identified a range of regulators. And I'm showing you highlighted here 2 regulators, CCP and COD-Y, that regulate different genes across a variety of different metabolic pathways. And this illustrates the importance of systems level approach to understand how these commensals might be modulating the phenotype of C. difficile. So using this model, we then predicted how you would have flux changes that would manifest differentially in the wild type or in the individual knockouts of CCPA or COD-Y, or in the double knockout of CCP and COD-Y in monocolonized or PBI co-colonized contexts. And this just at a high level shows you how the individual knockouts have their own unique effects. But when you have the double knockout, you have a different set of effects that change completely in a co-colonized context, right? In a co-colonized context, you see flux through a lot of critical pathways are reduced in the pathogen in C. difficile when PBI is present, right? So based on that, we predicted that individually, each of these knockouts would not have significant consequences on C. difficile infection, meaning PBIs will be protecting them. But if you knock both of them out at the same time, that would worsen the infection. And through mouse studies, we then validated this. We essentially showed that individually, neither of the knockouts on their own have appreciable effect on infection outcomes, but the combined knockout has a synergistic epistatic effect that manifests only in the co-colonized context with PBI. So I will stop there, but quickly summarize that I've shown you 2 models that give you some very unique capabilities of going from nucleotide resolution to phenotype through regulatory and metabolic networks. So the EGRIN model, for example, allows you to take compendia of transcriptome data and reverse engineer for any organism on context-specific regulatory networks. And we can, in these networks, associate regulatory mechanisms down to nucleotide resolution of promoter motifs, even in intergenic regions. We can also predict in what context these regulatory mechanisms act and how they change the combinatorial association of genes in different conditions. And furthermore, we can predict the consequences of perturbing the regulators at a transcriptional level. Now with the prime, we can predict how these consequences go on to perturb flux through metabolic pathways and how that manifests in phenotype, in particular growth rate and biomass. And we can set the objective function to a range of different things and predict effects of transcriptional regulation on all of those downstream consequences. This is quantitative because we're using weighted influences of regulators. So we get quantitative phenotypes which can be rank ordered. And I showed you with a few examples that they match up beautifully with the experimental determined fitness values. And we can uncover also systems level mode of action of drugs and gene knockouts that allows you to depart from that one drug, one target kind of mindset, because we all know drugs have off-target effects and global effects. So with this systems approach, we get much better understanding of the global effect. In fact, there was a paper published that a lot of drugs, 90% of drugs, act by mechanisms that are different from what we had believed them to be. So this kind of gets at really understanding how drugs act. And finally, based on knowledge of how these individual drugs act, we're developing new approaches to predict which drugs would act synergistically or antagonistically. So in the end, you can not only identify new vulnerabilities and drugs that target them, but you can also identify combinations in which the drugs would act effectively. So I'll stop there again. Quick acknowledgement for all. All right, so I'll stop sharing.
Okay. So the floor is open to questions for both of our speakers. In a sense, we had one bacterial talk, one viral talk, but in fact, there's a lot of commonality. The computational methodologies are different. A lot of the questions that they raise are rather similar. Go ahead. Okay, please go ahead.
Hi there. Yeah, cheers guys for both the talks. I wanted to ask Daniel a couple of questions about his multi-scale model. That's okay? Perfect. I remember when you were showing it on one of the first slides, there was the, you showed that the transmissibility in the sort of between host model was a function of the pathogen load at the within host scale. I've sort of been reading up on different multi-scale models as part of my lit review for my PhD, and don't tend to find a lot of data for specific diseases how these are linked. So I wondered, for this model, was it a sort of an assumed linear or logistic relationship, or was it actually calibrated to data? Like how did you determine the relationship between pathogen load and transmissibility? So that was the first question. Sorry, yeah.
Oh yeah, if you want to ask both, that's fine.
Oh sure, yeah. Yeah, I guess the second question was, in terms of running the whole model, was it that you sort of assumed that within host one was in a sort of equilibrium state and then use that to run the between host one, or was there an algorithm for choosing which of the scales to run at each time step?
Yeah, okay, great. Oh, thanks, Lucas. So yeah, first question, it's somewhat data driven because it was somewhat tuned to epidemiologic data. So I can put the paper in the chat afterwards, but it uses the serial interval of both influenza and SARS-CoV-2 to sort of differentiate the contact network a little bit. I would say it's pretty weakly data driven though, as far as like compared to the viral load modeling, which is tightly data driven. And the second question, the viral load stuff happens sort of on top of the multi-scale model. So there's really it's sort of a stochastic simulation where you assume that there's a person who's infected on day one, how many people do they see, and what's their viral load, and then if any of those people get infected, their viral load starts, and their viral load parameters are sort of drawn from the heterogeneity of possible theoretical people that we've seen by fitting to a bunch of viral loads. So then I think it's cool that we got a lot of the same results you would get from a sort of more simple analytical model, even though it was this pretty asynchronous model where you have person one infecting 5 people, all their viral loads start, one of them infects somebody else, that viral load starts. There's depletion of susceptible people, so there's a little bit of competition baked into it. But I hope that answers your question. So it's very literally programmed, I guess you could say.
Yeah, excellent. Yeah, cheers. Thanks, Andy. And yeah, the paper link would be really useful as well.
Great. Yeah, totally. And if you want to email me later too, just please do.
Excellent. Yeah, cheers.
Okay, so questions from Lucas. Did you assume a certain interaction structure in the stochastic epi model? Yeah, so there's probably some underlying temporal network. So there's no like network. And part of the reason we did that is just because at that point in time, we were really wanting to be careful about like not stigmatizing certain people as the super spreaders, like as if that's their personality. I think it's that's tricky, because if there are people that tend to go out more often, it's sort of, that's their phenotype. But I think as a result, again, the sort of the way it was implemented is that there is people that see other people, and the number of contacts for person A is drawn from this gamma distribution that's roughly data driven, and the number of contacts for person B is roughly gamma distributed. So at each time point, you redraw the number of contacts for a given person, and there's no correlation within person, let's say, between their contacts. So I hope that answers that question. And two, do I know what's driving the evolution of SARS-CoV-2 in immunocompromised patients? Is it related to clearance rate, or have similar observations been made for other viruses? So I don't know the answer to that yet. I think that's sort of like the question maybe worth trying to model. And what I will say so far is that if you impute the typical within host evolution rate of SARS-CoV-2, it's very low. So most people with SARS-CoV-2 infection have like maybe one mutant one base pair mutation by the end of their infection, sometimes zero. So it's pretty slow within host. And if you then say, what if there's somebody who has SARS-CoV-2 for 100 days, like some of these people have this prolonged infection, that's maybe enough to explain the amount of divergence that's seen in those studies, but I think not quite. I think there might be extra selection pressure potentially from having a weak immune system rather than like a good immune system or no immune system. You can imagine there's some situation in which having a high viral load for a long time with some directed selection pressure can push evolution faster. But this needs to be like fully modeled, I think, before I can give you a good answer on that.
I want to follow up on that there, because and it's actually not unrelated to the second talk to me, which is when you do evolutionary modeling, it's always tricky, because when you have mutation, as we saw in the second talk, it can move you very far from where you started. But typically we assume some kind of diffusive mutation, diffusive evolution in our parameter space. And that diffusive evolution in parameter space may or may not correspond to the actual possible space of evolutionary jumps that are achievable. And so you mentioned sort of as a primary consideration, the evolution of your effective reproductive number. But as you point out, the reproductive number is a function of all sorts of different behaviors of your virus. And so I was wondering how you actually implement the evolutionary strategy. How do you decide how do you actually do evolution? What parameters evolve? Because the R is an emergent property of the virus. It's not an intrinsic thing. And so that was my first question, was essentially how do you actually do evolution so that it looks something like real biological evolution, which isn't diffuse? Or do you do not? Are the other stochasticities of your problem enough that that's not an issue?
That's such a great point. I would frame this as well as the difference between like genetic evolution and phenotypic evolution. You can have a one base pair change that does a huge phenotypic change, right? So those are not necessarily coupled, and particularly in that HIV model, I did not couple them directly at all. So I don't have like a bit string that's mapped to some phenotypic landscape either. So for the COVID epi model, it's implemented through viral loads. That is what is assumed to be the sort of like biological evolution, is that it gets a higher peak viral load, and that sort of maps to a higher transmissibility. Right. It doesn't change your contact patterns, like it doesn't. Right. But we explored a little bit what happens if you change the like average number of contacts, and of course that makes a massive difference. So if you get a virus that evolves to effectively make you see more people, meaning like, maybe, you're just spewing farther into a room or something, that could make a big difference.
Or if you're infected and you're asymptomatic for longer, so that you're going around spreading it for a longer period of time before you get sick.
Indeed, yeah, and we have no isolation.
Was that one of your parameters in your model, that the eclipse phase, basically, or the latent phase, when you're infectious and asymptomatic, so that the duration over which you can spread changes? Is that an evolutionary parameter?
Not in this paper. We sort of like these two parallel stories, which is this more multi-scale, slightly, I guess, like coarser model, and then we have this purely epidemiologic model, but we put multiple variants into it. And then in that case, we do change. We did test whether perhaps Omicron has a shorter, eclipse phase, and that does help explain. Particularly if you saw this, Omicron upslope was way faster than some of the other variants. Yes. And that's, a parameter that can really help that. But we don't have the combination model where I can get to that level of epidemiological parameter granularity. I think that would be a good meeting point, honestly.
I mean, we had this wonderful talk. The second thing, which I think ties the two talks together a little bit, was we had this wonderful talk by Tanover a few weeks ago. And one of the things he showed was how incredibly effective SARS-CoV-2 is at inducing interferon-induced antiviral resistance in secondary tissues. So he could get a primary infection in the liver, a primary infection in the lung, a primary infection in the kidney, anywhere he wanted. And the interferon response was so strong that it almost never spread. He almost never got major infection in any other organ. Now, if you're immune compromised, you could imagine that one of the things that could happen is you could get a lot more loci of secondary infection. And as we just heard in the second talk, if you have a lot of heterogeneity in locus of infection, you have very different selection pressures. And so I wondered whether one possibility, and I wonder if people have looked at that, I suppose they have, is whether one of the things that could be happening in your immune compromised patients is that you're getting infection in other tissues, and that spread of the virus to secondary tissues could be leading to different selection pressures that could be leading to rapid increase of heterogeneity of the viral genotype.
That's a really interesting idea, and I don't know of any models off the top of my head that have tried to model. I mean, surely in epidemiologic context, spatial and evolution, but within host spatial evolution, I think is just growing now.
Because one of the variables I don't think was included in the tuberculosis talk we just heard, although we heard it a couple of months ago in the talk that Kevin James gave, not on SARS-CoV-2, was temperature. And some viruses are very temperature sensitive, others are not. If you have Omicron and it's staying primarily in the nasal and respiratory passages, so it might be temperature adapted to a lower temperature than a Delta, which is more lung focused and is going to be acting at a higher temperature. And so that could also be another mechanism of variation. If you're in the testes, if you're in some peripheral tissues that have a typical lower temperature, you could then be creating another evolutionary drive that could strongly select for different viral genotype. I don't think you even have to have a lot of virus to do it. You could have a small population of the virus somewhere else that could be driving that rapid evolution.
Yeah. So overall, I think anatomic data is just more and more crucial to help understand this.
And I apologize, I talked a lot. Let's come back to, is that Rahul who had a question?
Yes, thank you so much. Both talks were, sorry. Thank you so much. Both talks were really fascinating. I had one question for the first talk. I wanted to ask is how does when you showed like super spreading, right? One way to think about it is like dispersion in the reproductive number. Have you considered other ways in which some models that don't consider super spreading explicitly might incorporate that kind of dispersion? For example, like environmental stochasticity in transmission, or like measurement error. Like how do you kind of disentangle those potential effects from super spreading? And then my question for the second talk was, when you look at for some of the pathways that you're able to identify using the techniques that you developed, how transient or static are those pathways, in the sense of, is the bacteria itself changing those? Are those pathways changing very quickly over time, or are they relatively kind of stable so that you can conceivably design drugs that target them?
I've talked a lot. Nitin, do you want to?
Yeah, sure, I can. Yeah, so there are both of those phenomena. There are some pathways are changing dynamically, and that's why you need time course experiments. And using the topology of the network, you can in fact model the time course changes in states of the pathogen or the host, whatever you're modeling, and get at transient vulnerabilities. We've done this in also in cancers. In glioblastoma, when cells undergo transition from proneural to mesenchymal state, with single cell analysis, we could track the trajectory. And that may also get to some of the questions that were discussed earlier. And these are a mix of both adaptive and evolutionary states, where you have gain of mutations, or you have transcriptionally driven or metabolically driven physiological state changes. But if you have the right kind of measurements and the right time samples, you can get at the transient states. And then you have the steady states, where typically flux modeling will give you steady state predictions. So by doing the integration with regulatory networks and getting time course samples, we can get at transient metabolic states as well.
That makes sense. Thanks.
Sure.
And just on the super spreading question, then, to be clear about our model, the dispersion is in the number of contacts, and then there's also variability in viral loads. So there is sort of two baked in types of heterogeneity. The noise question is kind of interesting too, because let's say for the epi modeling, which I didn't present today, we're always fitting to like 7-day averaged or something like that. And I mean, if you look at these data, you can see what happens on the weekend versus the weekdays. There's like big fluctuations in caseloads just by reporting. So that is real. And I think the only way we can get around that is by smoothing it out and sort of going through the middle.
That makes sense. Thanks.
I mean, unfortunately, one of the lessons of your modeling is that masking and social distancing are the best way to prevent new variants from showing up.
Yeah, well, vaccination.
They cut both of your.
Yeah, it's true. And I mean, if you make assumptions about how much masking, let's say, reduces the effective transmission probability, it really seems like most people that spread COVID are doing it at peak viral load. And if you lower that a little bit, then you can really lower transmission. So there's strong assumptions built into it, but the whole story basically shakes down to non-pharmaceuticals being really important. Vaccinate as fast as you can, and then wear masks.
We had that nice talk about Mexico and how festivals were great super spreaders and promoters of diversity of viral genomes. And so that was an interesting one. And of course, from Fred Hutch, Schiffer's talks about some of these issues have also been very informative that way. So it's great to see these things put together in that context. I guess I wanted to come back to the bacterial. The pathway mapping you're doing is incredible. It's sitting on the back of a lot of perturbative experiments. And I was trying to think about what it would take to design a set of experiments to begin to do the kind of pathway mapping that you're doing for tuberculosis, or you mentioned now for glioblastoma. Where do you start? In other words, you're sitting on, I mean, tuberculosis has had an enormous amount of perturbative work done. And you showed your microreactors and the variables you're changing. It's a bit intimidating for somebody thinking about designing a set of perturbative experiments to actually start mapping pathways. What's your advice for somebody who's coming into that? I work with a lot of, for example, toxicologists.
Yeah.
And so a lot of the same questions come up: combinatorics, non-additive effects.
I'll answer it in a couple of parts. So one, for metabolic modeling, you don't need any data. You can, as you know, you can do reconstruction just from the genome sequence. You do need to do some validations. Those could be at a phenotypic level, but you don't really need a lot of molecular data to build your first metabolic model. For the transcriptional network, you do need a few hundred data points, more the better. And for TB, we had 2,000 or over 2,000. But this used to be a bigger problem in the past. I mean, I'm not saying it's easy, but it's definitely a lot easier to generate these data sets now, because you can do multiplexing, and you need to pick a dozen conditions or less. And for each of those conditions, and these could be changes in temperature, pH, and you have to pick perturbations that are ecologically relevant to that pathogen. And then you need to do profiling in 2 dimensions. One would be dosage responses, and the second would be time course responses. And each of those experiments will give you a column in your matrix file. And so when I say a few hundred conditions, it includes both dose response and time course measurements. And so we've done as few as 100 or less transcriptome profile data sets. We built reasonable models. The models definitely get much more accurate with hundreds and thousands of conditions. And now we've done this for dozens of different dozens of different organisms. And to our surprise, if you go to public repositories, there's a lot of data that have been generated by a variety of different groups. What we did for TB, 53 studies, 30 groups. For, of course, cancers, you have the TCGA, but for a variety of different pathogens, the community has contributed data sets that are compiled in a way that you can use. And we've done this for yeast by compiling, not doing a single transcriptome profile experiment, and building a model that was predictive. So I think it'll take a couple months to build a model, including experiments.
I mean, in your case, I mean, your example was tuberculosis, but then you mentioned doing it in glioblastoma. So that's a much bigger genome if you're in the host. And I guess that's the question too. Do you think that there are, I mean, how important is it to actually understand the regulation going on in the stromal cells around in your granuloma?
Yeah.
I mean, you're focusing on changes in the TB, which certainly seems to be sensible, but presumably the cells around them are responding to the infection as well. It's a very heterogeneous environment for them as well. And
you definitely, I mean, we have also, as I said, done eukaryotic network modeling, and we've done integrated modeling as well. And it's possible to do. Where it becomes challenging. Say in glioblastoma, we did it with bulk tumor profiling from patients. And this was from the TCGA days when they weren't really sorting tumor cells, and they still don't do it. But we could still develop a predictive model that could take this mass of tissue that was predominantly tumor cells, but you also have normal cells and many different cell types. But you can still get signal because you're integrating over multiple patients. The common unifying factor across patients is the cancer, right? So you do get signal, but with single cell techniques, we are now building single cell networks, and then you can get at subpopulations of different cell types and the networks that are active in those subpopulations. We're also now slowly building single cell methods to monitor host pathogen interactions as well. And that way you can get at host cell subtype specific networks on both sides, on the host side and on the pathogen side, and how they're communicating. So there are ways you can get at it. I think the bottleneck is good technology to make the right kinds of measurements. But those are coming online.
So what would an example be of a particular thing you need to measure that's difficult now that's coming online?
So for example, there's chromosome accessibility with things like ATAC-seq. You can now do, well, until recently, you could do it only on bulk samples, but we can now do it at single cell level, both RNA sequencing and ATAC sequencing. And in fact, there's a newer technology that is being developed here in UW and the Hutch that will allow you to ATAC-seq and RNA-seq on the same cell. So what that will allow you to do is find out what is a chromatin accessibility map within a given cell type, and what is the expression pattern within that cell type. The two pieces of information together will get you at mechanistic drivers of regulation.
Okay, time for a last question or two. Lorenzo, you usually ask a question.
Well, not really my field today. I don't have really big ideas. I obviously mine was probably would have been what you do with the karyotic cells about inferring pathways, but it's already done.
Well, is it done sufficiently? I mean, are you confident that you know the pathways in your delta notch regulation enough? Couldn't there be other things that are going on in your endothelial cells that you're missing?
No, I'm sure that, well, actually, I proposed once to actually do transcriptomics of endothelial cells, just perturbing the endothelial cells and try to infer the pathway. These are the probably, I don't know if the tools can be translated directly, the bioinformatic ones, directly from bacterial biology or prokaryotic to eukaryotic cells, if we have to include some more complexity.
Yeah, no, I think even the network inference for eukaryotic cells, so we have a different way we do it. I think there's the approach we call signal for systems genetics network analysis. There we do some of the same things by clustering like we've done with the prokaryotic cells, but because we can't do cis-acting regulatory influences in a straightforward way, we take ENCODE data, and we've generated a database of transcription factor binding locations that are associated with chromatin accessibility and footprint. So there's more evidence that the transcription factors may be actually binding to certain elements in certain cell types. And same with microRNAs, we can do reverse engineering of microRNA regulation by looking at conserved 3' UTR binding locations. We can do that de novo, or we can look at a reference database, and there's a framework we developed to do that. So conceptually it's similar, but mechanistically it's different in how we infer it. And that's why we were able to do this with glioblastoma, multiple myeloma, and different more complex regulation in eukaryotic cells.
Great, thank you. I mean, I was thinking specifically of Lorenzo's experiments when I asked the question, which is why I wanted to kick it to him a little bit. But other last question, we're at our time. Well, I want to thank everybody. That was wonderful, 2 wonderful talks, and I'm glad we had a bit of time to talk about it. It was very stimulating. I hope you can both come back. There was clearly a lot of material that you had, you could have presented in addition to what you talked about today. So maybe come back and present other projects or drill down in more detail. I think we would benefit from hearing more detail about the approaches, the computational approaches for both projects. And thinking a little bit more about how to measure things. I know that's a very vague thing to say, but it's a topic of some interest. How do you quantify, especially longitudinally quantify states of living organisms, whether it's at the molecular level, the cell and tissue level, or whole organism level? Those both come up in both talks. So again, I want to thank everyone again for a great panel, and I look forward to seeing our regular people again, some tomorrow and the rest of you next week. Thank you again. Bye.
Thank you.